Okay, Calem Kenward is a UBC, are you a postdoc? PhD student, rather,
PhD.
trainee. And
Yeah, if you go back one slide, it'll give all the info.
yeah. I don't know why it's not going one back slide. Here it is. Here we go. Fine, we got it. Okay, he's a student in Natalie Stranaka's lab, who has a huge group working on COVID, as she's a structural biologist, well known throughout. And Calem is working in her lab. So Calem, you're on.
Hi, everyone. Thank you for joining today. And so I'll be sharing something a little bit different. I'm hoping to give a brief look into how we as structural biologists are helping to provide insight into SARS-CoV-2. So at this point, everyone is obviously very familiar with the COVID-19 pandemic and the massive global effort to study the virus. In contribution to this effort, our lab is aiming to characterize the atomic structure of various mutant proteins and in an effort to validate AI predictions and also gain insight into the function of the molecular machinery behind the virus. So the culprit of the COVID-19 pandemic, SARS-CoV-2, belongs to a diverse lineage of single-stranded RNA viruses. These viruses normally circulate in bat reservoirs, but are capable of spilling over into other mammalian species. Notoriously was the recent spillover into pangolin and then eventually humans. And so the overall virulence cycle shown here is fairly straightforward and can be roughly divided into cell entry, replication, and then viral release. And each one of these steps involves many different structural and non-structural proteins. Ed, can you advance twice? Yeah, because I accidentally put some boxes in there. So my research focuses on the machinery involved in some of the early steps of the cycle, highlighted with the red boxes here. But today I'll just focus on the study of the main protease of SARS-CoV-2, or MPRO for short. But the approach I'm presenting here is fairly representative of our attempts to characterize many other protein targets. So after the virus has entered the cell, the genetic material of the virus is translated into a long polypeptide, which is shown in step 2 here. Ed, can you advance? Yeah. And the genome of SARS-CoV-2 codes for many different proteins, including various structural components, such as the spike, envelope, or membrane proteins, as well as several non-structural proteins, such as the RNA polymerase or viral helicases. All these proteins are initially translated into long polypeptides, which are then cleaved by internal proteases such as MPRO, which interestingly also cleave themselves from nascent polypeptide. Can you advance, Ed? So the catalytic cycle of MPRO involves several different transition and intermediate states. I'm not going to get into this to too much detail. I just want to highlight that there are distinct residues that are critical for this proteolytic activity, namely a catalytic cysteine and also an adjacent histidine in the active site. Recently, the Stranaka lab published the first structure of the acyl intermediate of MPRO, shown here. And this was done using a catalytic mutant to trap the protease in this state. So the colored cartoons represent the structures described in that paper. I'm hoping to examine some of the other structures, the ones in gray, and further characterize this process using a histidine 3304 to alanine catalytic mutant with the goal of trapping the cleavage target in the active site. In this case, our target is the C-terminal auto-processing sequence that MPRO would cleave to free itself. Next slide, please. To do this, I've chosen 3 different lengths of C-terminal extension to study. I've prepared fusion protein constructs which contain additional protein tags just to help with expression and purification. So in this case, a hexahistidine and a SUMO tag. So each of these constructs were transformed into E. coli, in this case BL21, and expressed. And we obtained multi-milligram amounts of MPRO. And so the overall purification process can be visualized with an SDS page gel, which just separates proteins in a sample based on size. So as the shown here in the gel, the lower it goes, the lower the molecular weight of the protein. And so just after the lysis, so it's total and supernatant, so shown there, it's pretty difficult to tell where the expressed MPRO is. But after purification using a nickel affinity column, which binds our hexahistidine tag on our constructs, we can see a band corresponding to the his-sumo MPRO around 48 kilodaltons in the first E lane there. Oh, sorry, can you advance once more? I know I forgot about the animation for that box. But then, very quickly, we cleave off this expression tag and do another round of purification. And in the end, we end up with a fairly pure sample of MPRO at around 25 kilodaltons, as we expect. And our final purification is done using size exclusion chromatography, which separates our proteins based on size. And the resulting trace here, we can see that a dominant signal peak corresponds to pure MPRO. Next slide, please. And so after we have pure protein, we next try to crystallize the proteins by adding various additive salts and precipitants. And so far I've had early promising success with each of the constructs actually. Just here I've shown a few examples of range and quality, and I'm still working on optimizing these crystals for the next step, which is X-ray crystallography, where where a diffraction pattern is generated from an X-ray beam passing through our target protein crystal. And we can use these diffraction patterns to generate electron density maps, which we fit our protein structure into. And we hope that these maps are high enough resolution and quality so we can model the structure of MPRO and gain some structural insight. So I know that was a lot of steps, but hopefully that provides some small slice of insight into some of the work that goes into just partially characterizing just the mechanism of just one SARS-CoV-2 protein. Thank you. Any questions?
Can't hear me, you can't see me. That was great, Calem, thank you. Are there any questions? Where's our questioners? So with this material then, is the plan to look at compounds that interfere or bind or in terms of therapeutics or for diagnostics? Do you know what the plan is?
Yeah, so for this one specifically, we're just interested in like further understanding the mechanism behind MPRO, and because there are many therapies that already target MPRO, and it is a promising target for both antiviral therapy and vaccination. For some of our other targets, definitely we are already looking at an antibody response.
So these are intracellular, though, by and large, you know, at this, I mean, are you going to be targeting, you think, intracellularly from a therapeutic point of view?
Yeah, some of the small, I don't know the names of the drugs off by hand, but some of the smaller molecule antiviral drugs already do target some of these proteases.
Okay, cool. Great. Okay, thanks, Calem. Appreciate it. All right, next speaker is, I'll try to do this a little better, is Imane Boukhatem. I hope I got that. Is that right? Oh,
yeah.
Good. Okay, I'm going to just, thank you so much. I'm going to just share your, so Imane is a PhD candidate from the University of Montreal. Supervisor, Dr. Marie Lord-Kipanidze. I'm sure I didn't do well on that one. Okay, but I'm going, oh, you're already sharing. Oh, good. Is that okay?
Yeah, perfect.
Great. Go for it.
Where's that? Okay, so hello everyone. Today I'll be talking about BDNF signaling in human platelets. Can you, next slide please? Thank you. So really quickly, the brain-derived neurotrophic factor has been discovered in the brain, hence the name, but it's also increasingly studied in the cardiovascular system. And what I think most people don't know about BDNF is that it's also stored in human platelets with very high concentrations that can reach up 1,000-fold those of brain levels. What's also really interesting is that when we activate platelets, say with collagen or ADP, there is an important release of BDNF from the platelets. So why is BDNF stored and released from the platelets? That raises the question to what is the importance of BDNF in platelet biology? And in our lab, we sought to investigate the effect of BDNF on platelet function. In the next slide, we found out that BDNF will induce irreversible biphasic platelet aggregation. So we use light transmission aggregometry, LTA, which is pretty standard in aggregation testing. And as you can see on the bottom left of the slide, we have aggregation traces. On the x-axis, we have time, and on the y-axis, we have percentage of aggregation. And it starts with a flat curve, and if we have aggregation, the curve will go down. And as you can see, while the vehicle and low nanomolar dose of BDNF do not induce platelet aggregation, medium and high concentrations which are still in the nanomolar range, will induce complete platelet aggregation. Also, if you look closely on the BDNF traces, so the red and green one, you can see that they have a biphasic profile. The first phase is usually slow, and this phase is usually when the ligand binds to its receptor and activates the platelet. And the second part is quicker with a much steeper slope, and this is usually the results of the secondary mediators jumping in, being released from the platelet, and inducing amplification of the aggregation. Since BDNF seems to be showing a biphasic curve, we wanted to check if BDNF relies on secondary mediators. So in the next slide, we checked by blocking 3 main mediator pathways. The first one is alpha-2 beta-3 B-beta-3 integrin, which we blocked using integralin. The second one is the COX-1 pathway, which we blocked using aspirin. And I guess the most important one is ADP, which we blocked through its receptor, the P2Y12, using RC, which is the equivalent of clopidogrel. And as you can see on the aggregation traces, on the blue curve, which is BDNF, inducing a nice aggregation, the use of the 3 inhibitors of the secondary mediators blocked BDNF-induced aggregation, confirming that BDNF relies on secondary mediators. We also wanted to know what receptor BDNF binds to induce this platelet aggregation. And of course, the first one that comes to mind is the TrkB receptor, which is a BDNF receptor in the brain. So in the next slide, we investigated the presence of TrkB on platelets since it has never been shown before. So on number 3, we did some Western blots of TrkB, and on the first line, we have our positive control, which is human cortex lysate. And as you can see, we have both the full length receptor and a truncated form of the receptor. But if you look at the last 4 lines of my Western blot, we have our human platelet lysates, which only express the truncated form of the receptor, which is pretty interesting because the truncated form in the brain lacks the kinase signaling domain and have a different signaling pathway, which I will come to in just a second. But right before that, we did some microscopy tests, and we used a brain cell line as a positive control, and our TrkB is labeled in green. Of course, our cell line expresses the TrkB, but if you can see in our wash platelets, they express TrkB both on the surface of the cell as well as inside the cell. Moving on to the last slide, we wanted to know which pathways BDNF will activate in platelets. As I mentioned earlier, we know that BDNF is stored inside the platelet, released from the platelet, and will activate the platelet. So if you move with me to the right side of the slide, we did a bunch of biochemical and functional testing that I'm not showing for the sake of time, but this is just a summary. So we found out that BDNF will bind to a truncated form of the receptor that will recruit a GTPase protein called RAC1. RAC1 will activate the PKC pathway known for its calcium signaling in platelets, and will also activate the PI3K pathway that will phosphorylate AKT and STAT3. Altogether, they will induce platelet aggregation and release of secondary mediators, as I showed you earlier. So in conclusion, we're still trying to understand the physiological role of BDNF in order to hopefully one day establish a link between platelet function and cognitive health. That's all for me. Thank you so much.
Can I ask a question? Ed, are you there?
Yes.
Only if you let me ask a
Wait,
question too.
I am here. It takes a while sometimes. Okay, yes, Wolfgang. Thank you very much, Imane, and go ahead, Wolfgang.
Okay, then I go first.
Yes.
It was a great talk. I noticed that there was a very, very long lag phase before these platelets go into aggregation, before the second wave meteors kick in.
Yeah.
I mean, what's the explanation for that? And does that correlate with RAC activation? If you do a kinetic study, does RAC activation come in late too, or what's going on there?
Well, we think that BDNF might not be classical agonists like collagen or ADP, but it could be a primer. I'm not showing any results here for the sake of time, but what we did was we incubated platelets with very low concentrations of BDNF that do not induce platelet aggregation, and we also incubated them with very low concentration of collagen or ADP. We did PAR1 and PAR4 receptors, and alone, they didn't induce platelet aggregation, but when we pre-incubate platelets with BDNF, they enhance the aggregation. So we think it acts more like a primer than a full agonist, if this makes any sense.
Which is actually similar to serotonin in some ways, which is another brain factor that is highly expressed in platelets and doesn't really activate platelets, but is a cofactor in some ways. So very interesting.
Yeah.
Okay, Wally, sorry, Wally, hey you, and then I have a question from Jeremy Wood. Wally?
So does BDNF, is that made in megakaryocytes and packaged in alpha granules, or is that taken up during circulation?
Such a good question. That's actually my PhD project I'm working on right now. There's two hypotheses. It's either internalized from the circulation or inherited from megakaryocytes. There's evidence in the literature for both, and both say it's either only megakaryocytes or internalization. So that's what we're working on right now. But it could be both, yeah.
Okay.
Hey you?
Yeah, very nice. So I'm very glad that you're working for the philosophy doctor, PhD project. So I therefore to ask you the function. So this described as a brain derived this factor and it has brain protective function, but that also could induce platelet activation. So can you understand why Mother Nature designed that for us?
That's a good question, actually.
No, not a big question.
So BDNF is known for its protective as a protective agent in the brain. It's also known to be in enhancing like factor in cancer cells too. So it's not only positive. And what's also interesting, something I didn't mention, BDNF has a propeptide called pro-BDNF, which will bind to another receptor called P75NTR. And P75NTR is actually known for its apoptosis effect. So we're not sure what is it doing exactly in platelet by activating them. And as I said earlier, answering the question, BDNF could maybe be like a cofactor in maybe, I don't know, in healing process, coagulation could be just released on some action site, and then so a good question. We still don't have the answer to that.
I'm gonna move on. Jeremy, are you there, or you want me to ask your question?
Yeah, I can ask.
There you go. Hi, Jeremy.
So my question is that patients with Huntington's disease, one of the symptoms of that is that they have decreased BDNF secretion in the brain. And so I was wondering if anyone has looked at whether platelet BDNF is also reduced in those patients, and whether there are any platelet phenotypes in those patients.
I'm not sure if this has been looked into. Actually, one of my colleagues presented the work this morning in a poster. He presented some work we did with cognitive disease, linking BDNF levels with cognitive disease patients. And we're finalizing this paper actually. And we have a small portion of patients. So we have a family with autism where we have BDNF level affected, and it's related to a mutation in BDNF. So I don't know if it's close, but this is some evidence with platelet function and BDNF mutation in cognitive deficits.
Okay, Imane. Oh, sorry, go ahead, Jeremy. You wanted to finish?
Oh, no, I was just going to say thank you.
Thank you.
All right, thank you, Imane. That was fabulous. Okay, we're going to move on to our next speaker, trainee speaker, Wayne. Wayne Zhao is a PhD student in Dr. Dana Devine's lab here at the Center for Blood Research, and he's going to be speaking about metabolomic analysis of cold stored platelets. Okay, so I only have to share and get you going.
Thank you for inviting me to this talk. I want to switch gear a little bit and talk about a different aspect of platelet biology relating to platelet component storage. So today I'll be sharing with you my thesis project on the metabolomic analysis of cold stored platelets. Next slide, please. Next slide. Thank you. Just to give you a little bit of a background for platelet storage. So now all of our platelets used for transfusions are stored at 22 degrees under constant agitation. Now this room temperature storage actually poses a significant risk of bacterial growth for transfusion recipients. It also directly leads to a very short storage time for our platelet components, only up to 7 days. So recently people have been looking at, re-looking at cold stored platelets. Because they have the potential to reduce bacterial growth and have superior hemostatic effectiveness. So to understand these cold stored platelets a bit further, we decided to look at the metabolomes of stored platelets. Next slide, please. So this is very much a collaborative project. So here in the CBS and the CBR, we obtained buccal platelet concentrates, which we used in a pool and split model to generate 3 identical bags of platelet concentrates. They have been stored. They were stored in 3 different conditions. So the first condition in gray, which I call RP, they are the standard storage condition. And then we have 2 units that have been stored at the cold temperatures. The first one under shaking condition, which I'll refer to as CPAs, and non-shaking condition, which I'll be referring to as CPs. We then sampled these platelets for metabolomic analysis on day 1, 2, 7, and 14. And we went over to the University of Colorado with our collaborators, Angel and Davide, which we ran some UHPLC and mass spec, analyzed the metabolite levels of stored platelets. Next slide, please. So to get into our data, because I can't use the mouse, I designed these labels for you to look at some of the important metabolites in the graph. So first, we looked at the glycolysis and the TCA cycle of our stored components. First of all, if you follow the half moon yellow symbol, that points to the glucose level during storage. And as you can see, the room temperature stored platelets consume more glucose than the cold stored platelets, beginning after 7 days. That's probably because their glucose concentration has been depleted. Next, if you follow the red heart, you can see that that points to GSH, which is the glutathione level. And now this metabolite is extremely important in defending platelets against oxidative stress. And what you can see is that the cold stored platelets have a relatively stable, slight decrease in their GSH, whereas the room temperature platelets in gray have a massive decrease during storage. And lastly, if you follow the green lightning bolt, you can see that glutamine level during storage. Now, glutamine, in this case, in room temperature platelets, it looks like the glutamine has been used as an energy source. And this relates to the TCA cycle, where it points to the room temperature platelets have high mitochondrial activity and high energy demand. Next slide, please. So when we're doing the metabolomic analysis, we also looked at some of the protein degradation markers. So we noticed that if you look on the right, under the red protein degradation markers, we noticed that in room temperature stored platelets specifically, there is an elevated level of acetylmethionine, which is a marker for protein degradation. And also, when we look at the methionine metabolism in general, we notice that there's an elevated amount of methionine in the room temperature stored platelets. Now, reactive oxygen species can interact with methionines to form methionine sulfoxide. And this, again, is an evidence that during room temperature storage, but not in cold, they experience high oxidative stress. And furthermore, If you look at the end of the pathway where acetylcysteine L-homocysteine is, SAH is the precursor for homocysteine, which can be metabolized in its own pathway to make reduced glutathione to defend against oxidative stress. Next slide, please. So finally, we did some correlation study with our in vitro function measurements and metabolites. So here's an example where we measure the platelets during storage using rotational elastography to measure their clot efficiencies. So here shown is the MCF, or the maximum clot firmness characteristics of the clots measured by ROTEM. Now we noticed that when we correlated the metabolomics data with the in vitro data using Spearman correlation, we see that metabolites of the pentose phosphate pathway, and that's shown in red, and high energy nucleoside compounds, they correlated positively with our MCF values. So this shows the importance of physiological energy and oxidative stress to cause strength. Furthermore, if you look in the green, that's the metabolites that are mostly negative correlating with the clot firmness, and that and most of the metabolites come from the purine catabolism pathway and energy expenditure. So our next step for this study will be to use biological inhibitors to modulate some of these platelet metabolism and see what happens when we store these platelets again in standard and cold conditions. Thank you.
Mute. All right. Questions. So it's interesting, the shaking is not, there's no detrimental effect of the shaking as compared to non-shaking in the cold situation.
Mm-hmm. Yeah, so for the future studies, logistically it'll be easier to have the platelets non-shaking, because if you were to shake again, you have to set up a shaker in the cold room.
Right. Gary, I can see you're unmuted, so you must have a question.
Yeah, myself, we've been looking at cryopreservation of platelets for a different purpose, and particularly for use in laboratory testing of procoagulant activity. And I'm just wondering, and we'll have a abstract at ASH where we found protocol for cryopreservation, which seems to preserve the essential procoagulant activity. And I'm wondering what you're finding. It looks like you focused on the mechanical contractile activity of the platelets. I just wonder if you've looked at the procoagulant activity.
I have in my previous study, in my poster lab. So because I only have 5 minutes with this presentation, I didn't show those data. So I did look at in multiple different models using aggregometry and using ROTEM with other different parameters to look at coagulability. We've also done some studies in vitro to look at using host platelets in a tabletop transfusion model as well. So we did look at that. Yeah.
Any other questions? Yeah, go ahead, Wally, please.
So have you looked at any lipid metabolites in your studies? It looks like you focused mainly on soluble metabolites. Did you look at any lipids?
Yes, we did do lipidomics. So those data, again, I didn't have room to show those data, but we ran a comprehensive list of over 270 metabolites plus lipidomics. And I can tell you that there is a significant difference in the acetylcarnitines level of these platelets. And also we noticed that the levels of smaller peptides, smaller lipids, they're easily depleted. They're more preferentially depleted in the room temperature setting. I think they're being used for energy again in the mitochondria. Right. But hopefully those data will be published soon and you'll be able to view them. We publish everything in our paper, so you should be able to see them pretty soon, I think.
Okay. All right, thank you.
Great. Okay, Wayne, thank you so much for giving your presentation. And we're going to move on to Wolfgang. Wolfgang, you're going to run it off yours, I assume?
If I can.
Well, you're doing a better job than I have, I'm sure. Okay, I'm going to introduce you, though, while you're doing that. Dr. Wolfgang Bergmeier is a professor of biochemistry and biophysics and a member of the University of Chapel Hill, UNC Chapel Hill Blood Research Center at UNC School of Medicine. Wolfgang studied biology at the University of Regensburg in Germany and graduated from Witten-Herdecke University, the Department of Molecular Oncology. Working in the fields of signal transduction, platelet biology, and hemostasis and thrombosis, he joined the lab of Denise Wagner at Harvard as a postdoc and was soon promoted to instructor in pathology. And there he studied molecular mechanisms leading to platelet damage during extended storage and to platelet adhesion at sites of vascular injury. He then moved to Thomas Jefferson University in Philadelphia, where he joined the academic faculty of the Cardozo Foundation for Hematologic Research, and he built an externally funded research program, again investigating signal transduction pathways critical for platelet function. In 2011, Wolfgang moved his research program to UNC Chapel Hill. His lab identified the small GTPase RAP1 and its regulators as a unique rheostat for platelet reactivity. His ongoing work focuses on a better understanding of small GTPase signaling pathways in platelets and megakaryocytes and how to translate his basic science findings to the clinic. Wolfgang, thanks so much. And the podium is yours.
Ed, I hope you guys can see this.
I can.
Good. So thank you for the invitation. This has been an absolute joy to be here today. I was particularly excited about hearing from patients, hearing from doctors, getting the whole picture from the clinical side of things. We heard about platelet transfusion. We just heard about platelet storage. We heard about the hemostatic activity, mostly tested in vitro. And I think my talk is designed to kind of bring that into an in vivo context. And so we use mouse models to study how transfused platelets and human platelets work in a more physiological context. And that's really what I want to get across with my talk today. Now, before I get to my stuff, just disclaimer. The one that is relevant for this is funding by Merck. So Merck funded part of the data that I will show, and I will point out what that is. Now, so my outline is give you a brief introduction on what we do and on platelets a little bit, then talk about the hemostatic function of transfused platelets in the context of qualitative platelet disorders. And then in the final couple of minutes, talk about the model that we built that I think is really great to study human platelets in mice, and especially the hemostatic and thrombotic function in a, as I said, a pathophysiological environment. Now, I can't believe that we made it to the end of this day and nobody really put up a slide on how platelets actually work in a hemostasis situation. So I'll do this here. They are obviously in a very unique environment. They are in blood flow. They have to adhere under shear conditions. And for that, I have a very special machinery. It's a multi-step process. They have a receptor on the surface called the GP1B59 complex, or von Willebrand factor receptor, that detects disruptions in the endothelial lining, parts of the extracellular matrix, von Willebrand factor being exposed there. And basically that's important to slow down the cells, make contact with the matrix, and then allow agonist receptors to engage at the site of injury. And for the first wave of activation, we are talking about thrombin receptors and collagen receptors, or other receptors for matrix components. Now these signals lead to activation of the cell. There's all kinds of changes going on, probably the most important one being activation of integrin receptors, the cell going from a low adhesive, a low affinity, a low adhesive state to a pro-adhesive state. And that allows the platelets to make contact with the extracellular matrix, but also to recruit more platelets into this growing plug. Now for the recruitment of additional platelets, we need second wave mediators. We just heard about those, ADP thromboxane, released from platelets working in an autocrine and paracrine fashion to sustain activation signals. Why is this all important? Well, because we can interfere with certain aspects of this cascade in order to prevent thrombotic complications. First drugs that were developed were integrin inhibitors, very strong antithrombotic activity, also very strong anti-hemostatic activity, not really used a lot anymore. We have a recently approved drug called Vorpaxar, PAR1 inhibitor, so one of the thrombin receptors. And I will talk about this in the final part of my presentation. But really what most people are treated with is aspirin and P2Y12 inhibitors, and that takes away the second wave of platelet activation and therefore the excessive growth of thrombi when they form. Now, my lab focuses really mostly on the events that lead to integrin activation. So this key event in platelet adhesion. And what you see here is the intracellular side, or the events happening at the integrin in a resting platelet and an activated platelet. In resting platelets, there is an enzyme expressed in the membrane that makes sure that there is no activation going on. We characterized this protein RESA3 a couple of years ago. If you don't have this protein, or mice that don't have the protein, are completely thrombocytopenic because of premature activation of platelets. In the case of activation of the cell, the integrins shift from a bent over low affinity state to a high affinity state. This is all mediated by an intracellular adapter protein called TALEN. TALEN binds to the cytoplasmic tail of the beta subunit of the integrin, causes separation of the transmembrane domains between the two subunits, and that leads to activation of the receptor. Now, a key player in this activation process is the small GTPase RAP1, and as Ed already said, this is the focus of my lab. RAP1 binds the TALEN and basically makes it activate the integrin. And in order for RAP1 to get activated, you need an additional protein called CalDAGF1, an activator of this small GTPase. CalDAGF1 gets recruited to the membrane, and then due to calcium binding, gets activated, activates RAP1, activates the integrin, and CalDAGF1 will be important in the first part of my talk. So remember CalDAGF1 as an activator of RAP1. Just two slides on two methods that we developed now a couple of years ago that I think can be really helpful to the field, certainly help us a lot in my lab. One is a different hemostasis model. Most people use the tail bleeding time model to study hemostasis. We wanted a closer look to also get a visual what's going on at the site of injury. And so what we do is we expose the saphenous vein in the leg of a mouse, and then we use a laser ablation system, basically a laser gun, to punch a hole in the blood vessel that's about 50 micron in diameter. And when we do this and we look down onto the blood vessel, we see platelets immediately filling this hole. There's a fibrin ring that actually surrounds the hole, and then on the right you have the merge. So the ring, the fibrin ring, we think holds the platelet plug in place. We can hit the same spot again after, in this case, 5 minutes. We get a second response that is a little stronger. Fibrin ring gets stronger, and then we can do it again. Third time, you can do it as often as you want, as long as you can blow out that plug from this position. Then, of course, you can go up and down the saphenous vein and make multiple injuries. So out of one experiment, you get a lot of information on fibrin formation, platelet adhesion, and bleeding times, which are about 15 to 20 seconds long in this particular model. Now, if you look at this in 3D, so this is scanning confocal imaging, what you see is the vasculature here, the platelet plug sitting in that area where we use the laser ablation system. And just to kind of orient you, how many platelets do you need to form the plug? This is differentially labeled platelets that we put in a mouse, and I'll get to this in a minute too. You can basically count across how many platelets are necessary to span that area. And we're talking about approximately 30 platelets across to fill up that hole. So this is a very small lesion, probably not really reflective of all the things we see in the clinic. In the clinic, we probably have bigger lesions, but a really great model to get images for how hemostatic plug formation happens. Now, the other model that we use a lot is called the adaptive platelet transfer model. And in this case, we have taken advantage of a transgenic mouse model that was created by Thierry Weir a couple of years ago, where he replaced the extracellular domain of the GP1B alpha subunit of the fibrinogen factor receptor on the platelet surface with the human IL-4 receptor alpha subunit. He did that to basically study what it means to not have this GP1B receptor, what it means for hemostasis. We realized that this is actually a great way to induce thrombocytopenia in mice, because what we do normally in wild-type mice is we use antibodies against the GP1B alpha subunit to induce rapid and complete thrombocytopenia in those mice. However, the same can be done with antibodies against the human IL-4 receptor in this transgenic mouse. So you put those antibodies in, and within minutes, the mouse is completely thrombocytopenic. There are no platelets left in circulation. And that, of course, has the advantage that you can now bring in donor platelets, transfuse those in, and since the donor platelets do not express the human IL-4 receptor, these platelets will not be targeted by the remaining antibody, and so you can reconstitute the mouse with the platelets that you're interested in. So you're generating basically a situation where the mouse is not altered, but the platelets are altered. And so we can do this, and depending on how many platelets we transfuse into the mouse, we can basically titrate in what platelet count we want to have for our studies. And this is really a great model for this first study that I want to share. This is work done by Bobby Lee in my lab, who was recently promoted to assistant professor. And I also want to highlight my friend Pepe Rivera from Spain, who was helpful with some patient studies that actually I will not talk about. So we got into this because of patients that were identified, and Pepe actually was one of the first ones identifying these patients that have mutations in this RAP1 activator called CalDAC-F1, this protein that gets recruited to the membrane. And the gene is called RAS-GRP2, so keep that in mind too. And so there are mutations along this gene, and basically all the patients that we have the same or a very similar phenotype. Moderate to severe bleeding, the PFA 100 is severely prolonged, but platelet count and clotting activity are normal. Platelet aggregation is impaired. Actually, these patients were initially characterized as Glanzmann-like, so very strong impairment of platelet function, but not really to every agonist. So some agonists, there's no aggregation, some agonists, there is aggregation. Now, what was unique when we looked at these patients, or what surprised us is the fact that in the treatment of these patients, often platelet transfusion seems to fail. And for somebody who doesn't really work with patients a lot, I was surprised by that, because I thought if you have dysfunctional platelets and you give healthy platelets, you should fix the issue, right? So we started thinking about this and started reading the literature. And as people already pointed out today, we really don't need a lot of platelets in circulation in order to maintain hemostasis. So normally in the setting of thrombocytopenia, one apheresis unit is enough to prevent bleeding. So these patients get one apheresis unit, everything is good. However, in qualitative platelet disorders like Glanzmann thrombasthenia, that doesn't seem to be the case. And this is a very nice example published by Lisa Jennings many years ago now, where they had a patient that got transfused with platelets before a procedure, and they gave him 1 unit of apheresis platelets, and that did not prevent the bleeding. And so afterwards, they looked into the ratio of dysfunctional to functional platelets, so GT to normal platelets, and the ratio was about 5 to 1. In order to prevent the bleeding, they had to come down to a ratio of about 1 to 1. They gave an extra 4 units to this individual. And I think that's often done in the clinic just to prevent any sort of uncertainty that there's still bleeding going on after treatment. And so we thought, well, we have good mouse models. Maybe we can get a handle on what kind of a ratio is required. And is this really also going on in the CalDAC patients? Can we use the mouse model to figure this out? So what we did is we used our RAS-GRP knockout mice, and we treated them with wild-type platelets just to see how many platelets do we need in order to prevent bleeding. And we used our laser injury model to get to the bleeding time, but also visualize what's going on. And so what you see here is the bleeding time in wild-type mice in black. So this is just 15 seconds, as I said. The dotted line up here is basically a thrombocytopenic mouse, where we eliminated all the platelets in circulation, and they bleed basically forever. So we stopped the experiment at this particular time point here. The RAS-GRP knockout mice, so the CalDAC-F1 knockout mice, they bleed to a similar extent, even though they have some adhesive function, but it's not enough to prevent bleeding from happening. Now, what we have to do in the thrombocytopenic mice is we only have to give a few platelets in order to get hemostasis back. So this is about 5% of what the normal platelet count is in a mouse. That is enough to bring hemostasis or bleeding times back down to almost normal. Now, the same is not true when we give the same amount, actually more platelets, twice as many platelets to a RAS-GRP knockout mice. So in the presence of these dysfunctional platelets, it looks like these transfused platelets are not as active anymore. And just like Lisa Jennings had to do in the GT patient, we had to give about 6 times as many platelets to get the bleeding time down to where it's supposed to be. All right, we thought, okay, we have this. What about if we use our adaptive transfer model? Can we go after what ratio do we need in order to, between the dysfunctional and the functional platelets, in order to get hemostasis back online? And can we visualize what's going on there? And so we did this study. And so what you see here is all these mice were rendered thrombocytopenic. So the endogenous platelets were removed, and then we gave wild-type platelets only, so bleeding time was low. RAS-GRP2 platelets only, bleeding time is long, just as you would expect. And then a 5 to 1 ratio of RAS-GRP2 platelets to wild-type platelets didn't really affect the bleeding time at all, even though we had enough wild-type platelets to really prevent bleeding in a thrombocytopenic situation in the presence of the RAS-GLP2 platelets. That was not the case. And again, we had to go down to a ratio of 2:1 or less actually in order to get hemostasis fully reinstated. So we looked at this by imaging too, and in this case, the platelets are differentially labeled. You will see the wild-type platelets on the left and the mutant platelets on the right. Up here, we have the 5:1 ratio, and what you see is that bleeding doesn't stop. So there's blood continuing, there's blood that continues to come out of that lesion, while in the 2:1 ratio situation, bleeding stops nicely. And what you see is that both platelets, or both platelet preparations, are actually incorporated into this hemostatic plug. So even though these RAS-GLP2 platelets have a severe defect in aggregating in vitro, in adhering in a PFA 100 situation, in the presence of intact platelets, they are just fine incorporating into this plug. Okay, so we knew that, so that there's something going on. So we thought, can we get more information by doing high-end imaging about what's going on in the plug? So what we did next is we basically selected one slice within the plug and looked at the platelets in the plug in that slice to see how they interact when we have a situation where we have wild-type platelets in different labels, or mutant wild-type platelets in different labels. The ratio is the same, so the 647 label platelets are 2:1 versus 488, but in the lower case, we have mutants to wild-type. And so when we did that, what you see is, or what you will see, is that in the wild-type situation, both platelet preparations go into the plug basically at random. That is what you would expect. So they are localized next to each other, and they are also very tightly packed in the plug. However, in the mutant wild-type situation, what you see is that the plug is very unstable. There's a lot of sloughing off of platelets at the edges. And not only that, what you see is that there are areas within the plug where the wild-type platelets are not present anymore. And what you see is mostly mutant platelets. And if you go in later on and we take an image of that, you can see that there are territories within the plug that are basically exclusively occupied by mutant platelets, and there are territories where we have mostly wild-type platelets. So these mutant platelets get recruited into the plug, but then they cannot really participate in the adhesion or in the aggregate formation, and so they get squeezed into certain areas where they become a liability for the stability of the plug. So that's one way that your endogenous platelets affect your transfusion efficiency. The other way is, well, the other question is, how do these platelets actually get into the plug in the first place? And the key to that is this initial adhesion step mediated by GP1B in binding to von Willebrand factor. As I said before, in this case, the platelet doesn't have to be activated as long as GP1B is there and von Willebrand factor is expressed. And it's not only found in the matrix, it's also found on the platelets that are adhering at the site of injury. These platelets will be recruited. And that's exactly what we see here. So we have a situation where we have wild-type platelets and wild-type platelets where we cleaved off the GP1B receptor. And what you see is that these platelets devoid of GP1B, they cannot make it into the plug properly. That means they also cannot destabilize the plug and they cannot impair the wild-type platelets in what they are supposed to do. So to sum this up, in a thrombocyte or in a normal situation, you have your platelets forming a plug, just as I showed you. If your patient is thrombocytopenic, you give donor platelets, they function just fine, and the endogenous platelets are normally not a problem because these platelets are functional. However, if you have a qualitative platelet disorder, this is not the case anymore. You have a lot of dysfunctional endogenous platelets. They compete for binding sites on VWF, and they also disrupt the stability of the plug. And what I haven't shown you, but we did similar studies for antiplatelet therapy because, of course, qualitative platelet disorders are fairly rare, but antiplatelet therapy is used a lot. And you have a similar issue there where the stability of the plug is just not very good anymore. So you would have a similar issue in that situation. All right, I have a few more minutes. So that was the study on transfusing platelets into patients with qualitative platelet disorders. But really what you want to do is you want to study how your human platelets work. And you can do this in vitro, but it would be nice if you can study them in vivo too. And as I said before, this is a study that was sponsored by Merck because they had this PAR1 inhibitor that they developed, but they couldn't really study it or test it in a mouse model because mice do not express PAR1, only human platelets express PAR1. And so they asked us if we could devise a strategy to basically do adoptive transfer of human platelets into mice, but in the absence of mouse platelets. So what normally is done is that you get volunteer platelets, and this is all published for many years now by different groups. They can be transfused into immunocompromised mice because these immunocompromised mice, they can accept xenogeneic grafts. If you do this in a regular mouse, these platelets are cleared really quickly, so you can't really study them. Now, so this happens. What you also have to do is you have to give some human von Willebrand factor because the mice don't really have the proper von Willebrand factor for the human platelets to work with, but all of this has been worked out. The problem is that most of these studies were done in the presence of endogenous mouse platelets. And as I just showed you, as long as there is adhesion to von Willebrand factor, well, if your mouse platelets form the clot, your human platelets will go into the clot and it will not tell you anything about the function of these cells. So the idea was can we do adoptive transfer in these immunocompromised mice? And so the first thing we did is we just repeated what other people showed, is that if you have a not-skid mouse and you put in human platelets, you can see those circulate for several hours, up to 24 hours. And really the window we need for our hemostasis model is about 3-4 hours. That's all we needed, and for that we have a stable platelet count. We can detect, of course, both human and mouse platelets by differentially labeling. Now, the problem we ran into is that when we depleted the mice, these immunocompromised mice, of endogenous platelets, we saw lethality that we never see in regular mice. And it's a long story, but in the end, the issue here is that there is an anaphylactic response going on in these immunocompromised mice, and you have to also give these mice a PAF receptor inhibitor to completely wipe out this response. So in the presence of a PAF receptor inhibitor, all the mice tolerate depletion of endogenous platelets, and now you can basically go from a pre-depletion, pre-transfusion situation to a mouse that only has circulating human platelets, no mouse platelets around. So with that on board, we did our studies, and this is just similar to what I showed you before. We did the 3-shot protocol that I showed you before. The platelets are going in just nicely. So platelets on the left, fibrin in the middle here, and then the overlay. So it looks very similar to what we find in mice with mouse platelets. The kinetics may be slightly different, but overall a very similar result. Having the system on board, we decided to look at some antiplatelet drugs that are used in the clinic. The first one we did is dual antiplatelet therapy, treating with aspirin and ticagrelor. And what you see is that platelet accumulation is reduced. Fibrin formation actually wasn't affected. And in this model, because the injuries are so small, you actually don't see a lot of bleeding in the presence of dual antiplatelet therapy. So we only saw some bleeding at the first injury, but then with more severe, with more fibrin formation, this wasn't a problem anymore. Now, really, we wanted to do this for the PAR1 inhibitor. So this is an analog of vorapaxar. And so we tested that. And what you see is the same thing. You get a reduction in platelet accumulation in the presence of a PAR1 inhibitor. In this case, we also saw an increase in fibrin formation that at this point, actually, we don't really know exactly where that comes from. And in terms of bleeding time, again, not much. There was a significant increase in the bleeding time in injury 2 and 3, but overall, very, very mild. However, the combination of the two of them, of the two strategies together, so it's the triple therapy with having dual antiplatelet therapy and this PAR1 inhibitor, we saw an even stronger platelet inhibition of adhesion of platelets. We saw a strong increase in fibrin formation, and we saw a strong increase in the bleeding time. So consistent with what people found in the clinical trials, giving PAR1 inhibitors on top of dual antiplatelet therapy is really not advisable. You get a lot of bleeding even in those small injuries that we created here. Just to show you what this looks like. So on the top here, we have the control. Down here, we have the triple therapy. And after the injury, what you will see in the triple therapy is that the clot actually, the plug forms, but then it opens up and the bleeding continues basically for the entire observation period. You dramatically destabilize the stability of this plug. And when we go in and do a 3D imaging, we see that these plugs look different. There's a lot of fibrin actually in the intraluminal surface of these injuries. And so with that, I think we have succeeded in putting together this adoptive transfer model. I think this could be a very, very powerful model for studying the efficacy and safety of antiplatelet drugs. But I think this could also be awesome for studying in vitro generated and stored platelets, platelet-like particles, and patient platelets. And with that, I just want to highlight again the two people who did all the work, David Paul and Bobby Lee. And thank you.
Fantastic work again. That was great. Okay, so I'm going to open this up to questions. Where's Hey You? Hey You always has questions, and I keep missing
Yeah,
them.
thank you.
There you go. Can't we see you? Hey You, where are you?
Yeah, Wolfgang, so you said that I'm very quiet today whether I'm sick. I tell you that I'm pretty healthy, and I really enjoyed your talk.
Thank you.
And also, I would like to let you know that recently I wrote editorial commentary published in JTH regarding this intravital microscopy. I did cite your paper. this model, okay? Carefully to read my reference, you will find that I cite well regarding your work with this model. So congratulations. I would think I enjoy your talk. There is no question, just the comments. Beautiful, and keep working harder.
Thanks.
I may provide some of antithrombotic reagents for you to test their function in this human platelet mice.
Sounds good.
All right. Wally, then Gary?
Yeah, so Wolfgang, that's really amazing. When you look at the mixtures of your wild type and your RASA knockouts, do you see changes in clot retraction if you measure that ex vivo?
So we haven't really done the mixtures, to be honest. In vivo, we see that these clots don't really, they don't hold on tightly, and they also don't seem to contract at all. So yes, I think we see it in vivo. We haven't done it in vitro, to be honest. RAB1 is important for clot retraction. We have studied that, but we haven't really done the mixtures of the two platelets.
Yeah, I'm just curious. Thank you.
But there's clearly, I mean, the way I picture that is that there is contraction between the wild type platelets when they hang on, but the mutant platelets cannot participate, and that is why they get squeezed into those territories where there is only mutant platelets, no wild type platelets in. But that's hard to prove, really.
Gary?
Yeah, really nice work. And it obviously, when something new is like that, it opens the door for so many more questions that could obviously be asked. I found myself wondering, like, what aspects in your model, what aspects of hemostasis versus what aspects of thrombosis are recapitulated there? Some of the things we're testing seem more like thrombosis, antithrombosis drugs, yet other aspects seem more hemostasis.
So the model is definitely a hemostasis model. There's nothing, I never claimed that this is a thrombosis model. We make a hole and there's a pressure drop, so the flow patterns change in there. So it's clearly a hemostasis model, right? The drugs we use to test the human model, the humanized mice, is just because these are drugs that are used in the clinic and there are some bleeding complications associated with the drugs. It was just a model system that we decided to pick for that particular story. But the puncture model is clearly a hemostasis.
Yeah, okay. I mean, you do get a ring of damage, like thermal damage from the laser injury. And right. I find myself wondering, is that to some extent like a thrombosis model?
So I think that Tim Stalker was on this early on. I don't know if he's still here, but I think they see the same ring of fibrin in their needle injury. It doesn't really matter what you use to make that hole. And that's actually an interesting point, is that it actually fits with the literature too. Denisa did those studies a long time ago in the mesenteric thrombosis model, where she used the fibrinogen knockout mice. And these mice, their only problem really is it's not the buildup of the clot or the thrombus, it's the anchoring of the thrombus to the vascular wall that is affected by not having fibrinogen or not having fibrin. And the same is what we see in our model, is that the fibrin is there to keep the plug in its socket, so to speak. It forms where you have the contact with the extracellular matrix and where you have the contact with the endothelial wall, so to speak. In the middle of the wall, there's nothing there to really activate the cells properly to cause fibrin formation.
And are you willing to speculate why adding the Vornapixor caused you to have more fibrin?
Yeah, I am. So there are two potential explanations in in the paper we wrote. Maybe it's the density of the platelet plug. Maybe there's just more thrombin diffusion into the plug. But since then, I just came across a paper published by Justin Hamilton at Monash University, where he looked at some of these precursors of these drugs and actually found that they had off-target effects that lead to a procoagulant response in platelets. And so I don't know if this particular analog that we got from Merck is one that causes a procoagulant response or not. I mean, that's easy to test. We just didn't do it at that time, but that probably is the likely explanation.
Okay.
But that also would tell you that having procoagulated platelets are important in the model to form fibrin, right? So that tells you where the procoagulant platelets are in the regular situation. They are around the plug in the intersection with the vascular wall.
Marie?
Marie.
Hey, it was a wonderful talk, as always. Thank you. A quick question. You know that distribution of kind of seed platelets and platelets that go onto it. So it reminded me of two things. So you did mention the core and shell model of Tim Stocker and that group, but it also reminded me of the work of Tim Warner that did the reticulated platelets and the older platelets. So have you looked at your possibility of just, you know, the newer platelets being transfused being different from the ones that are there? Is there any chance of that being part of the explanation?
So, I mean, the way this works is that we draw the blood from the mice and immediately give the platelets back to the recipient mice. So there shouldn't really be a difference in how old these platelets are. It's not like we have them stored for 5 days in a bag, which we should do, which would be fun, right? But so in this case, I don't think there's a difference in the age distribution between dysfunctional and functional platelets. I would say no. Overall, clearly, I mean, the younger platelets seem to be more reactive. Maybe these are the ones that are, you know, more powerful. Like if you look at Tim's work together with, I'm blanking on his name, the guy in Arkansas, Wally.
Yes. Yes. Yeah.
So anyway, they did.
Yeah, Brian Story.
Yeah, Brian Story. Wally actually just showed. You may show the data in a second.
I will.
So you see that the edges, again, is where the activation happens for most of the platelets, where they secrete, where they, you know, get fully activated. There are some in the middle too, but overall the edges is where most of the activity is. And it makes a lot of sense because you have to picture this like an open pipe, right? So the only interaction of the platelets with the wall is at the edge. In the middle, it's all soluble mediators. It's all ADP, thromboxane, maybe that's thrombin, maybe some brain-derived neurotrophic.
Could be, who knows?
But it's the wall where most of the high power activation is going on. And you need these immune type receptors that bind to collagen, that bind to maybe protoplanin to actually activate these cells enough to get PS exposure, to get full secretion going on. So it makes a lot of sense to me in the hemostasis setting. If you have a thrombosis setting, maybe everything is quite different. And that's the next thing we want to do is, you know, have an imaging platform like this for a thrombosis model too.
Thank you. Very interesting talk. Thanks.
I think you're muted, Ed.
Ed, you're muted.
Yeah.
Yeah, that's purposely done. Okay, thanks. Thanks, Wolfgang. I really appreciate it. Any other questions before we move on? No. Okay, thanks, Wolfgang. Excellent. All right, Wally, are you sharing your own screen?
I am.
Okay, you can start. I'll introduce. So Dr. Sidney Wally Whiteheart, as we know him as Wally, has been on faculty at the University of Kentucky for more than 25 years. Wally earned his doctoral degree at Johns Hopkins University School of Medicine under the mentorship of Dr. Gerald W. Hart, after which he was a postdoc scientist at Princeton University and Sloan Kettering Research Institute under the mentorship of Nobel laureate James Rothman. Wally's research focuses on the molecular mechanisms of platelet function, hemostasis, and innate immunity, and no one knows more about platelets and their granules than Wally. His group has produced more than 130 publications. He's well funded by the Department of Veterans Affairs and the NIH, serves on numerous grant review panels throughout the world. He was a two-term member of the editorial board of JBC. He's currently an editor of journal Platelets. He's appropriately being honored with several prestigious awards, including the University of Kentucky Alumni Association Great Teacher Award. Indeed, he's the director also of the University of Kentucky College of Medicine's MD PhD training program. It's a great pleasure to wrap up our program today with a presentation by Wally. You're on.
So first of all, Ed, I want to thank you for the invitation. This has been a wonderful day. I've learned a lot. I've seen some great science. And I'm sad that it's going to drop off here at the end for my talk. But I really appreciate Wolfgang sort of setting the stage for some of the things that I'm going to talk about. So I want to start off with sort of the laboratory philosophy that over the years is we tend to think of platelets as cells and as cell biological entities with both exocytosis and endocytosis and autophagy and all of these kinds of things. And so what I want to do today is I want to talk about the exocytosis part, the outie part of the cell biology. And so this starts with the platelet secretome. We sort of had a couple of discussions and talks today on the various things that platelets can release. Platelets have 3 different classes of granules. Nominally, a dense granule population that contains small molecules like ADP, serotonin, polyphosphates. They contain a more complicated class of granules called alpha granules. And these are interesting in that they contain a number of different proteinaceous components. Some of these have obvious functions. They're involved in adhesion, fibrinogen, fibronectin, thrombosbondin, von Willebrand's factor. And then some of them actually allow the platelets to manipulate the site or the microenvironment at a damaged site. These are things that are growth factors. The most famous one, platelet-derived growth factor. But then there are other chemokines and cytokines that actually can have pro and anti-inflammatory functions. And so there's a number of things that platelets can release into their extracellular space that can manipulate or change that extracellular environment, be it at a wound site or actually be it at a contact site with an immune cell. And then finally, platelets have secretory lysosomes. And this is something that's common to hematopoietic cells in the sense that they can release hydrolytic enzymes into the extracellular space. Things like beta-hexosaminidase, cathepsins, that are degradative enzymes. Now, the role of these is really still not completely clear, but perhaps it's involved in clot remodeling or something along those lines. But the question I want to sort of try to address today is, how does platelet cargo actually come out? And I want to focus sort of the first part of my talk on a couple of points about platelet exocytosis. And one of them is, you look in this micrograph here, there are a couple of features. You see something down here that looks like a cloud of cargo or a cloud of proteinaceous material. And this EM, the darker material, is protein because it's stained with the heavy metals. And you also see a structure up here that looks like it's a granule, but it's dissolving or decondensing. And this is cargo perhaps being dissolved and coming out, or in the process of coming out. And so we wanted to sort of better understand the process of platelet secretion to get an idea of what it does in hemostasis, but from a molecular standpoint, what's actually going on in the platelet. So a lot of our old kinds of analysis centers around data from slides like this, or data from experiments like this, where we take platelets, we stimulate them with various agonists, and then we measure the rate at which cargo comes out. And we use a marker for dense granules, serotonin, a marker for alpha granules, platelet factor 4, and I'll come back to that a little bit later because we use several other markers too. And then finally, beta-hexosaminidase is a marker for lysosomes. And using this kind of data, which is a time course, and you're looking at the percent release. So that's the amount of material in the platelets that actually has come out into the extracellular space. You can actually fit this to this function, and you can develop what we call a KEX value, or it's a rate of exocytosis value that can describe this curve for serotonin release, or this curve for platelet factor 4 release, or this curve for lysosome release. And if you look, there is a bit of heterogeneity in these rates of release experiments, depending on the various agonists that you use. And we're using saturating doses of each of these agonists here. Okay? So that makes one point. Different agonists can make a difference in the rate at which cargo comes out of platelets. Now, that also holds true if you stay inside of the same agonist. So here we're just doing a thrombin titration, but at each of these points, we've measured the KEX value, the rate of release of our different cargoes. And the point I want to make here is that the amount of agonist also makes a difference. So you can be out here where you're saturating the system, the agonist is overloading the system, or at least saturating the system, and you're getting the maximum response. Cargo comes out as fast as it can come out, right? And then in here at the lower doses of thrombin, you're at the range where it's the most responsive. a small increase in agonist leads to an increase in the rate of release. So the concentration of agonist actually controls the rate at which things come out. And again, this is serotonin, platelet factor 4, and beta-hexosaminidase. Okay. Now, we can look more in depth at the cargo that's being released and look at the composition of the releasate. And in this experiment, this is just a presence-absence. Did we detect a molecule in the releasate or did we not? We use antibody arrays for this kind of experiment. And it allows us to follow about 30 so molecules that are released into the extracellular space. And you can see thrombin, which is the strongest agonist we use, pretty much everything that we measured comes out into the extracellular space. A weaker agonist, R4, for the human platelets, you see some molecules are actually not expressed. They're too low to be detected in our array data, so their levels in the releasate are actually too low. So it tells you that the different agonists can lead to different levels of release of these different molecules. And again, there's a dependence on strength of agonist and complexity of the releasate. Okay. Now, if you take this data and you go one step further, we can measure the rates at which all of these molecules are released. And when we do that, we can plot those on histogram. And this was where we got a surprise. So when you think about secretion, most people know about neurotransmission, and that's what's called burst release, where a synaptic vesicle comes up and fuses at the synaptic bouton, and all the neurotransmitter gets released out in a bolus. Okay? And in fact, that's sort of naively what we were expecting. But when we looked at the rate of release of all of the different cargoes I showed you on the previous slide, what we found is that they partitioned out into 3 different groups that were characterized by these 3 overlapping Gaussian curves or Gaussian distributions. Okay? We had a fast set of cargo, things that came out really quickly, and serotonin is one of this class. We had sort of a slow class down here that came out relatively slowly. Beta-hexosaminidase would be an example of that class. And then all of the rest of the molecules sort of fell along these 3 Gaussian distributions. We had an intermediate class and slower classes. So this sort of surprised us because it suggested that platelet cargo is coming out at a controlled rate, and something is actually having an effect to control that distribution of cargo out into the extracellular space. Okay, so to sort of lead into where I'm going with this, we came up with a few observations of platelet secretion heterogeneity. First of all, dense granule release, serotonin, other molecules that are in the dense granules is the fastest, and it's also the most sensitive to the lowest concentrations of agonist. So this is the first thing that comes out. And this is fitting because ADP is one of those cargoes, and that's an important signaling molecule for the second wave of signaling that Wolfgang talked about. Alpha granule cargo actually has the most diversity in the release rate. There were alpha granule cargo proteins represented in all 3 of the classes that we saw in our analysis. Lysosome was the slowest, and also lysosome release actually required the most agonist for release. So it's the most insensitive to stimulation. Now, these were all activation dependent, as I showed you. The rate of release is directly proportional to the level of stimulation the platelets have. There were rate differences amongst the alpha granule cargo, which I talked about before. And one of the things that was perhaps also a surprise is that there was no thematic pattern to what we saw in the release. Basically, the mixture of cargoes were relatively random, and we didn't see molecules that were pro-inflammatory or anti-inflammatory coming out at different rates or in different groups. It was a seemingly random distribution. So we started asking the question, what factors affect these release rates? We made these observations, but what are some of the explanations? Because again, it's not like synaptic release or synaptic vesicle release. It's got to be some different mechanism with which cargo comes out of granules. And so in collaborations with Brian Story, who was mentioned by Wolfgang, and Richard Liebman at NIH, we've undertaken a series of experiments using serial block-based VM. And what this allows you to do is to build a 3D model of a platelet at EM level resolution. Okay? And that allows you to see a lot of the ultrastructure in platelets. And what I'm showing you is a single plane from that kind of analysis and a 3D reconstruction where we're focusing on an alpha granule and its contact to the plasma membrane, which is the green structure here. And there's two things of note here. One is this plane sort of demarks the opening or the pore at which the granule has now fused and cargo is actually coming out, right? And the interesting thing is there's only one. There's only one release point here. The other interesting thing about it is the granule actually is quite swollen. This is a much larger size than a granule in a normal resting platelet would look. So there's a swelling component. The other thing I want to point out that foreshadows something I'll talk about a little bit later is there's also this material, sort of a fibrous type material that's left inside this granule. So we think that this is a decondensing granule where the cargo is dissolved and actually come out of this single aperture or this single fusion pore. Okay? So that's one way that perhaps granule release can be controlled. Another way is something that we also noticed, and this is a process called compound fusion, where you see one granule fused to the outside, and you can see this is the pore leading in, and then you see another granule fused directly to it. And these two granules, and this is represented here in this single plane of the reconstruction, you can see that's the exit out, that's one granule, and then that's the second granule, these fusing together. This kind of sequential fusing, where this primary fusion event occurs, where cargo is coming out, and then there's a secondary fusion or compound fusion that occurs, further adds complexity to the release kinetics. Okay? So using that kind of analysis, we can actually measure the diameter of the fusion pore and see whether that changes during the exocytosis process. And I'll call your attention first to the measurements of wild-type platelets. So they were stimulated for 90 seconds or 300 seconds. And then we did the serial block face analysis and basically counted fusion pores. And since it's a three-dimensional reconstruction, we can get the diameter of the fusion pores. And at the initial stages at 90 seconds, you can see the distribution of pores. The average is about 100 nanometers or so in size. Over time, 300 seconds, you see the pores actually expand. So there's a dilation of the fusion pore in the wild-type animal. If we manipulate the secretory machinery that's required for the membrane fusion events and we drop its levels down, we can actually delay that fusion process and make it actually more heterogeneous, and altering the opening and perhaps altering the flow of cargo coming out of the granule. And then finally, we can delete most of the secretory machinery that's responsible for this, down to about 90% or so. And in this particular case, we see pores form, but there are not enough of this fusion machinery available to form stable pores, and so they don't last, and you don't really count any at 300 seconds. But what this tells us is that the fusion pore actually dilates, and the fusion pore dilation dynamics is actually controlled by the number of membrane fusion machinery that's available in platelets. And so to sort of summarize a lot of this, that we've come up with this sort of scheme by which granule cargo comes out of an alpha granule. You have the granule docks in a particular position, the membrane fusion machinery engages, the fusion pore is initially formed, and you have buffer extracellular fluid coming in to help dissolve the cargo. The granule or the pore opening expands, dilates. You see more decondensation or dissolving of the cargo, and you start to see secondary fusion events or compound fusion events that ultimately lead to release of cargo into these sort of clouds that I showed you in the one of the earlier micrographs. This is accompanied by granule swelling, And this leads to a controlled release of cargo and perhaps explains some of the kinetics that I showed you in the earlier slides. Okay. So this is one way where membrane fusion and influx of media into the granule can control release. But what about things inside the granule itself? Are there molecules inside the granule that can control this process? And I draw your attention to this sort of bottle brush type molecule that we drew in the granule here, because this is a protein called serglycin. This was originally reported by Barbara Schick's lab. Donna Wolf was the first author. This is a proteoglycan that contains chondroitin sulfates. And it's one of the only intracellular proteoglycans ever reported. It's found in different granules, and of course, it's found in platelets. It is a fairly small molecule, 158 amino acids, and it contains this long stretch of serines and glycines that are highly decorated with carbohydrates, specifically glycosaminoglycans. And we think it serves as a matrix for the granules. And I'll show you some data to try to support that. But one of the interesting features of this, and this was a very simple metabolic labeling experiment where we took megakaryocytes from wild type or serglycin knockout animals. We just incubated it with S35 sulfate to label the sulfated glycosaminoglycans that would be part of the heparin sulfate, the chondroitin sulfate structure. And one of the things that was a surprise is that serglycin is one of the major sulfated species in megakaryocytes. We are actually not expecting that, but it's one of the larger, one of the dominant species that we saw labeled with S35 sulfate. Also, to realize proteoglycans run generally as smears due to the large amount of glycosylation that they have. And of course, this structure was sensitive to the enzyme chondroitinase, confirming the fact that chondroitin sulfate was present there. So what we have is a large polyanionic molecule that's inside a granule. Now, we confirmed Dr. Schick's original observations and showed that this serglycin molecule or proteoglycan co-localizes with platelet factor 4, von Willebrand's factor, and P-selectin, indicating that it actually is in the alpha granules, which they originally reported. We saw no localization with LAMP1, which is a marker of lysosomes. So we're pretty sure that it's in alpha granules. The next thing we wanted to look at is, does it make a difference in granule decondensation? And so if you look here, serglycin, or wild type platelets and serglycin knockout platelets have the typical discoid shape in resting conditions. And basically the granules look pretty much the same. When we measure them, they're pretty much the same size. So there's no real difference, plus or minus this proteoglycan. However, when you stimulate the platelets, and again, I'm showing you the data for 300 seconds, and down here is the magnification. You look at the decondensing granules for wild type, and they look pretty much like what I showed you earlier. There's a fibrous material inside the granule, and the granules are swollen. In the knockout animals lacking serglycin, the granules have a much more granular appearance. And I'm sorry for that pun, but it is a darker staining, indicating there's more protein still left in the granules, suggesting that serglycin is important for solubilizing some of this cargo and allowing it to release. But remember, it's a polyanion, so it could serve as a counter ion for a number of these cargo. Okay? The intensities, or the difference in the intensities between the granules, is actually quantified here. And we also measured the size of the granules and showed that the lack of serglycin affects the swelling of the granules as well. So you're not getting the same degree of swelling of the granules as you get in the wild type platelets. Okay. So with all of that, does serglycin affect secretion? And so we turn back to some of the same experiments I showed you earlier. We looked at release from dense granules, and of course, serglycin is not in dense granules, so we didn't expect to see any effect, and we don't. Serotonin release from dense granules is the same in wild type and knockouts. There was no co-localization of serglycin with lysosomes, so again, we didn't expect to see an effect, and there was no effect on hexosaminidase release. However, there was a robust effect on platelet factor 4. Okay. Now, I want to make 2 points here. This is percent release. Okay, so the amount of platelet factor 4 that's still present in the seroglycin knockout platelets just does not seem to come out to any great extent. You can see that here is the knockout compared to the wild type. Now, there is a packaging defect, and I will perhaps go through this if somebody asks the question, where seroglycin does affect the packaging of platelet factor 4, but there's enough to measure release. So that particular molecule is not coming out to the same extent as it would in wild type. Now, the granules are fusing because we see P-selectin exposure, and that's because this granule membrane protein can actually exteriorize out to the surface. So we know the fusion pores are being made correctly, but this particular cargo is not coming out correctly. What about other cargos? So we looked at a couple of others. One, Rantes, which has a high isoelectric point, so it's positively charged. And Rantes has a little bit of a defect in cargo packaging, but no real defect in secretion. So the Rantes molecule comes out just fine from the granules. Its rate of release is pretty much the same as wild type when seroglycin is deficient. Interestingly, though, another protein that we looked at, another granule cargo called osteopontin, which has a low isoelectric point, meaning that it would be negatively charged, actually comes out faster. And in this particular case, you can see that the knockout actually has a much faster, more extensive release than does the wild type, suggesting that perhaps seroglycin is playing a negative role in the release of this osteopontin molecule. And when you remove it, it actually is released more effectively. So this sort of allows us to look at this model a slightly different way. What are the various things that can control how cargo comes out? Well, one is the rate of fusion, okay? And that's probably directed by the degree of activation that you see, because several of the molecules involved in secretion are calcium binding proteins and sensors and would respond to platelet activation. But there's also a biophysical control, a fusion for its opening or dilation, the dissolving of cargo, and this polyanion seroglycin that's inside the granule is also playing a role in selectively solubilizing some cargo and actually not solubilizing others. And so we think all of these are contributing to that complex platelet secretion kinetics that I showed you in the very early slides. And so each one of these things is a contributor to the process. Now, why would this be important? Well, one of the probable reasons is that these cargo molecules have to have an effect locally. Clots are formed in a blood vessel. It's a flow cell. Things are moving along. And so if cargo is released as a burst, it's actually going to be flushed down the system and have no effect on the microenvironment. Whereas if you have these kinds of controls where you can, in a sense, platelets become a slow release aspirin, they can release some of these molecules so that they can have an effect and not be washed away by blood flow. Okay. So to sort of take the next step in this, we wanted to actually understand the heterogeneity of secretion inside a blood clot. And this was something that was alluded to in the previous talk. This is work that's been done between Brian Story's lab at Arkansas, Tim Stalker's lab at Penn, who's now at TJ, and the Richard Liefman lab. And basically in these studies, and what I'm showing you is a jugular vein that's been punctured with a needle puncture, okay? And so you're seeing the blood vessel wall here and the blood vessel wall here, and this is a cross section through the middle of the clot. And to orient you, this is the luminal side of the blood vessel, and flow is going in this direction. Now, there's a couple of things to note. One is there is extracellular deposition of platelets on either side. And the platelets are scored based on their degree of secretion, okay? Remember, these are EM images, and so each single platelet that's involved in this process can actually be drilled down, and we can determine whether they have released all their granules, some of their granules, or none of their granules. And so everything in red are platelets that are largely degranulated. They have actually obtained that maximal secretion rate that I talked about at the very beginning. And you can see a lot of those are at the downward side, or the downwind side, if you will, of the clot, where there's a lot of red platelets that are down here. Also draw your attention, though, there are a number of them inside the middle of the clot as well that are actually completely degranulated. The ones in green are platelets that are in close contact, that are making interplatelet contacts, but have not released their granules. And the ones in blue are platelets that actually still have their complement of granules and are loosely adherent. This would be thought of as the shell in the core and shell model, and you could think perhaps this is the core, though I think it's a lot more complicated than that based on the heterogeneity of the platelets. Okay? So this kind of heterogeneity of granule phenotype in the platelets led us to ask the question, when and where is platelet secretion important? What role does platelet secretion play in this thrombus formation? And so in order to address this question, we made a series of animals in which we have manipulated the platelet secretion machinery so that we can, in a sense, titrate down the levels. And I have to show you this one old slide here because it tells you the molecules that we're manipulating. We're going after the molecules that are on the granules that form a complex that bridges the two bilayers and mediates the membrane fusion event so that cargo is released. And these are proteins called vesicle-associated membrane proteins, or VAMPs. Platelets contain 6 of them. However, 4 are dominant. The other 2 are only present in trace amounts. So VAMP2, VAMP3, VAMP7, and VAMP8 are the dominant ones in platelets. And they form a complex that spans these two bilayers and mediates fusion. These are called snares, snare proteins, and there are some on the target membrane that bind to the VAMPs to mediate this process. And I won't talk so much about the ones on the target membrane. But we made these series of mice, and you can sort of look here in this diagram. These are the animals that we created with the VAMP proteins that are there, and of course the ones that are remaining. And when you look at the mass of the VAMPs remaining, you can see how we sort of titrated down the VAMP proteins. Whereas this animal here, which is a knockout of VAMP 2, 3, and 8, is actually also hypomorphic for VAMP7. And so we've basically taken away roughly about 90% of the VAMP proteins that are present in platelets. Okay? So this gives us a way to manipulate the secretion process. Have we really? So I'll show you some of the data. So when we delete VAMP2 and 3, nothing much happens. That's the green line compared to wild type platelets in the red line. Okay? Dense granule, alpha granule, lysosomes really not affected. When we delete VAMP8, which is one of the major VAMPs, we see a deficit in dense granule release, perhaps more of a deficit in alpha granule release, and a deficit in lysosome release. And the triple knockout that's also polymorphic for VAMP7, we see an even greater deficit in release, but not complete, right? There's still enough of secretion to go that's still occurring, especially for the dense granule. So I wanted you to remember these. The VAMP8 animals have roughly, if you want to use it here, about a 50% deficit in alpha granule release, maybe a 30 or so % deficit in dense granule release, and the VAMP238s, which have roughly a much greater deficit, maybe a 70% or greater deficit in alpha granule release, because these are the 2 animals that I will focus on in the rest of my talk. So we wanted to ask, do any of these have hemostasis defects? We chose 4 different models. We didn't have Wolfgang's wonderful microscopy model, so we had to go with the things that we could do. And so we took the animals that I showed you that have these varying degrees of secretion defects, and we ran them through 4 models. The tail bleeding model, which is the classic transection model. We used a jugular puncture model with the needle, and that's because of the micrographs that we have. We used a ferric chloride, a carotid injury model, which is an occlusion model, and then in collaboration with Rob Flamenhap's lab, we have some data on the pre-master laser injury model. Okay? So the question with all of these models is, under what context is platelet secretion really important, right? So for the tail transection model, basically there's not much phenotype. This is wild type. These are the various deletion models. None of these had any great effects on secretion anyway, so we expected them to look like wild type. When we get down here, that VAMP8 deletion animal, which started to have a secretion defect, really doesn't have a bleeding phenotype, right? I call this 50% secretion, but you saw the data and you can evaluate. That's not enough of a defect to have an effect on bleeding in this model. However, the triple knockout that was hypomorphic for VAMP7 has enough of an effect. And so that amount of secretion is not enough to actually form a clot in these particular models. And these animals failed to form a clot in the 10 minutes of the experiment that we used. Okay? So in that model, there's a window somewhere between here and here where there's enough secretion for clotting, and then not enough for clotting. So somewhere between those two. When we look at the venous thrombus model, this is the puncture model. The interesting thing here is none of the animals really had a phenotype. I'm not showing you all the ones that look exactly like wild type. I'm just showing you the ones I focused on. But the VAMP8 animals that really didn't have a phenotype in the tail bleeding assay didn't have a phenotype here. And the 238 animals also didn't have a statistically significant phenotype. So in neither one of these cases was there a phenotype suggesting that even this little bit of secretion that's available in these mice is enough to stop bleeding in this particular model. Again, a venous model, a puncture wound, much like what Wolfgang was showing. Okay. What about some of the other models? So in the carotid artery model, this is an occlusion event. Now we start seeing phenotypes with the VAMP8 animals. And so what this suggests is, like before, the triple knockouts actually don't form an occlusive clot because the 30% of release is not enough to form a clot. But now the 50% release, the VAMP8 phenotype is also not enough to form a stable clot like the wild type animals, right? So this degree of secretion is important in this particular model to form an occlusive clot. Now, finally, in the pre-master laser injury model, and I don't have a full data set for this, I'll have to show you what we have. But the VAMP8 animals actually do have a phenotype here. A clot forms, but it's slow, and it doesn't grow to the same extent as does wild type, suggesting that secretion actually is important for clot growth in this particular model, but it still can form even though it's slightly delayed. So these kinds of data sort of allow us to think about secretion as a potential therapeutic target. And there may be a magic window that's somewhere between that 50 and 30% release where we can manipulate the size of the thrombus so that we don't get an occlusive thrombus, but yet not have a bleeding phenotype. So we're thinking that somewhere between the VAMP8 nulls and the triple nulls is the sweet spot to target for inhibiting secretion and for control of thrombosis. Okay. So does this actually mean anything in terms of controlling secretion as a way to control thrombosis? Well, turns out nature probably has done part of the experiment for us in the sense that VAMP levels have been associated with cardiovascular risk. There's 4 papers now. 2 of these are GWAS experiments where VAMP8 levels, or VAMP8 single nucleotide polymorphisms in VAMP8 and microRNAs that control VAMP8 levels are correlated with increased cardiovascular risk and increased early onset myocardial infarction, suggesting that higher levels of the VAMP proteins may make platelets more able to secrete, or more efficient in their secretion, and thus driving their ability to form thrombi. Okay. All right. So with that, I'm going to sort of summarize some of the places that we perhaps can target platelet secretion. What I've focused on is the area down here where the membrane fusion events are driving fusion and cargo release. And I showed you how sarglycan and this pore dynamics are actually perhaps contributing to the rate at which cargoes come through. But there's a whole host of molecular protein-protein interactions that are required to get the granule into the right place to engage the snare machinery to mediate the membrane fusion event. And several of these actually offer opportunities for targeting. The exocyst and RAL complex can be targeted with RAL-specific drugs. This is work done by Alistair Poole's lab. One of the snare proteins is modified, is phosphorylated by I-kappa B kinase, and there are a number of different drugs that can be used to target that. And there are several protein-protein interactions along this pathway that might also be targeted to give us a way to manipulate the secretion process. Okay. And so finally, the people that are responsible for doing the work. The lab, I've been blessed with some wonderful colleagues over the years. The work I showed you with the VAMP animals is from a talented postdoc, Smita Joshi, and the work with sarglycan is from an equally talented postdoc, Harry Chanzu in the lab. And again, this work would not be possible without the collaborations with Brian Story and Richard Liebman's lab. Okay, and I'll stop there.
Thank you, Wally. You didn't disappoint. Can you take off your slides, please, so we can all see you? And we're open for questions. There's lots of questions, I'm sure.
Maybe I start, even though I just had Wally here at UNC.
Yeah, sorry about that. I tried to tell a different story.
So Wally, you said that there are options for targeting that machinery, but I guess the one thing that I don't know about is how shared is this machinery between all the secretory cells? I mean, including neurons and, neutrophils, inflammatory cells. I mean, how specific is this machinery for platelets?
Well, the interesting thing is that a lot of this machinery is shared with immune cells. So some of the proteins I didn't talk about, syntaxin 11, MUNC18B, and MUNC13-4 are actually all defective in a disease called hemophagocytic lymphohistiocytosis. And that's a cytokine storm disease. And so basically trying to target those might get you into problems with regulating the immune system. The I-kappa B kinase actually is an intriguing one because that phosphorylates a protein called SNAP23, which is important for forming the snare complex for that last fusion event. And there's about 18 or 19 drugs out there now that target that particular enzyme, and most of those have been safe and non-toxic. So that's one that we're actually interested in. The I-kappa B kinase knockout mouse we can use as a tool. It bleeds, so we know we've got a good target there if we can figure out the right drug. Your point is well taken, though. A lot of the machinery that we have discovered over the years is in immune cells, and if we manipulate too many things, we're liable to manipulate that as well, and that might be detrimental.
Hey, you?
Yes, Wally.
Yes, sir.
Very nice to see you, but Ed blocked me, so you're probably not able to see me.
Did I block you? I don't think so.
Yeah, you blocked me quite a bit of time, so that's even leading to Wolfgang say I'm sick. So because I use the way raise a hand, you did not see the hand, but see the chat. Yeah, so Wally, that was beautiful talk, and as always. And
thank you.
I believe that you understand the argument before between the Brian and the other group regarding the selective release of the cargoes of the alpha granules, right? I'm not sure whether you agree with them or you disagree with them. And if you agree with, I did heard that you used a selective release during your talk. So if you believe that they do have selective release of this cargo, So what's the mechanism could be, and so whether I have another naive question. Have you ever to see the cytoskeleton protein within the alpha granules?
So let me ask the last question first. I don't think I've ever seen with inside the granule. I don't think we've ever seen cytoskeletal proteins inside the granule. I mean, there are autophagosomes and things that could have cytoskeletal proteins in them, but no, I don't think they're in the alpha granules per se, or at least it's not obvious. To answer your other question, I mean, you know, this is something I see Joe's on there, so maybe we can talk about this some more. Is that I think platelets are capable of differential release when they're stimulated differently. You know, one of the problems, and I wanted to try to make this point, if you look at the kinetics of release at different concentrations of thrombin, right? Mm-hmm. I can make differential release by how I stimulate the platelets, right? I can make certain things come out more efficiently depending on how much I stimulate the platelets or not. And so, you know, is this a way that platelets interpret the signals that they get and give you a little bit of release or a whole lot of release? I think there's something to that. And these are, you know, we tend to think of platelet activation as this monolithic, you know, boom, they're blown out. And I think our work with toll-like receptor signaling and secretion from downstream of that is telling us that that's really naive. And there are different gradations of platelet secretion depending on the amount of agonist and even the agonists themselves, which ones we're using. So I think there's a lot more to it than this monolithic platelets just burp everything out. And we're now developing the tools to get at that subtlety. Mm-hmm. Sort of answer your question?
No, not completely, but my man.
what else?
Okay.
Hey, you. What else?
Visit him later. Let Joe jump in.
Okay. Thank you, Wally, for your effort. Anyway, yeah.
Okay.
Wally, I got a question for you.
Yes, sir.
Do you think any of these mechanisms of granule secretion that you're seeing in platelets are happening in the giant platelets and the megakaryocytes? Do you think the parent cell is using any of these to maybe, you know, secrete in the bone marrow and modulate that environment?
Ooh, yes, absolutely. The data that we have from the serglycin mice would actually argue that that's true. Those megakaryocytes from those knockout mice actually release platelet factor 4 in the media, and that controls megakaryocyte growth in culture. So absolutely, I think that this is one of the things that you and I need to have a beer and talk about, because I think a lot of granule biogenesis may actually be things going out and then coming back in through an endosomal compartment and packaging into alpha granules that way. And we've got some data with platelet factor 4 that's true. I know Morty Pons has got some data also suggesting that's true. So I think that platelet granule biogenesis is probably a lot more complicated than that, going out, you know, and being packaged de novo. I think there's a fair amount of traffic, retrograde traffic, coming back into the net. So yeah, I think that there's definitely more to do.
I have a question if there's time.
Please.
Who's that? Cool. Go ahead, and then Marie.
Hey, I hope you guys can hear me okay, but first off, it was a wonderful presentation. It's pretty funny because I'm actually presenting some of your data later this week, so I think you just scooped me, but that's okay. But my question is that there's been some studies performed, specifically EM studies by Van Nippen in the Netherlands, and he proposed that there are subclasses of alpha granules that are released, some being more tubular and containing multimeric von Willebrand factor, for example. I'm wondering if there's different subclasses that you found, and if so, how do they fit into your model?
can I speculate, if you'll allow me to speculate? I think that a lot of the things that we've thought are alpha granules may actually be sort of endosomal compartments that have got cargo that's been brought in and packaged. And so, some of the heterogeneity perhaps that we've all been observing, and Joe's observed, maybe because you're looking at different packaging intermediates. But one of the things that has shocked us, and I didn't talk about the endocytosis work, but it's been known for a long time that platelets endocytose, but I don't think people have appreciated how much they endocytose. If you look at the rates at which they bring in cargo, it's actually fairly significant. And so I'm thinking, we're beginning to think of alpha granules as some sort of specialized endosome, more so than a granule species. So just to shortly answer your question, I think you're looking at granules with different degrees of things that they've actually taken out. And that might be the best explanation for all of the data that everybody sees.
Marie, last question.
This was a wonderful talk. Thank you, Wally. I really appreciated it. This made me think of a paper from, I think it was Eric Bollard's group, who published the serotonin uptake from the gut and then release into the brain and then just go back circulating happily. And I have a hard time in the lab separating out the aggregation, secretion, and activation phases of platelets. So do you have an idea of what could be making them secrete without necessarily changing their cytoskeleton and aggregating? What could be a stimulus that we could look at in the lab that could separate out secretion from the rest of the aggregation cascade?
We've been looking for that kind of thing. I mean, you've got doses. I don't think that it becomes how much of a particular agonist you use. One of the things that I will say that's come out of our recent data, if you stimulate with toll-like receptors, and we've been focusing on toll-like receptor 7 and 9 and using agonists for those, we see a very different secretion profile when we use those agonists. The ATPP paper we just came out with, they don't really stimulate dense granule release very well at all, but they stimulate alpha granule release. So that's the only one that we've ever run across that's been a clear yes/no answer. I will have to say, and this is, dense granules are poised to release. They're docked on the plasma membrane. We know some of the machinery that's involved in that docking process. And so they are actually ready to go, and very small amounts of stimulation are needed for them to come out. There's more things involved in getting alpha granules and dense granules out, more proteins involved. But I think that's, if you look at some of the results with Hermansky Pudlak animals and other patients, that inability to release dense granules is a lot more important for hemostasis than the inability to release alpha granules. So I think there's a couple of things at play. Maybe I didn't answer your question, but there's a couple of things going on there that are different between those two classes.
But it is very interesting. Thank you.
Wally, thank you very much. Okay, with that, it was a fabulous talk. It was a fabulous day. With that, we're going to wrap up. Please don't leave just yet. Normally, I would be able to offer you wine, beer, cheese, food, and socializing immediately after. But we do have just a couple of little things just to tidy up before you go. So hang in there 1 sec. I'm going to pull onto my slide and see if I can do this. I haven't been doing so well today, I think. Try this. Okay. Do you see a picture of Earl? I hope you all do.
Yeah.
Great. Okay. First, I know, I know that Earl would have loved this. He really relished these kinds of events. He came all those years to us, year by year. He enjoyed the students. He enjoyed the talks. He enjoyed the patients in particular. He liked the wine. He just thrived off this. And it was such a joy to he came the night before. We'd go out for casual dinner. For those, many of you enjoyed that with us. We'd go out for Thai food with a whole pile of people, usually with Eddie Fisher at his side, and sometimes with his wife. And then with all the students, and then we go out for a nicer dinner the second night, and it was really a very, very special opportunity for me, for everybody who met with him, and quite frankly, I got to meet spectacular scientists from all over the world, young and old. So it was very, very special. So you can, I think you guys all, everybody, all the speakers, the students, the poster presenters. Everybody really lived up to what he would have really enjoyed. So kudos to everybody here. Now, we will have just a couple of more remarks on that, but I first want to hand over the mic to Stephanie. And Stephanie is here. Yes.
Yes, just to reiterate what Ed said, just want to give a special thanks to our poster judges as well as our trainee presenters for participating in the poster competition. I believe everybody did a really great job just from skimming through all the judges' comments. And the winners will be announced in just 1 second. These winners will receive prizes from BioRender. These are subscription prizes where they will get to make some nice figures for their research talks and publications and further conference posters. So with that, Ed, the winners are, we have 2 runners up, Nancy Yang and Andy Ann. And our first place winner goes to Jean-Christophe Bélanger. So congratulations. I will follow up with all 3 of you afterwards about the next steps. So congratulations. Back to you, Ed.
The quiz. All right, so the quiz was, before science, what did Earl aspire to be? I had various, I actually had a different answer. Dominique Chung suggested one thing, but I had a different question, answer. And what was Earl's favorite ice cream, and what did people in the lab call Earl? So this, when I visited Earl on his 90th birthday, and this is what he suggested. He wanted to be a pilot, but Dominique tells me that he actually wanted to be a jazz musician. So I'm not sure if Jim Jim Davie, I see you in the present here. Jim, have any idea what your father wanted to be? You'll have to unmute.
So I'm sure the answer is that he wanted to be a jazz musician.
Okay.
He also aspired to be a professional basketball player, but learned that he was too short.
He was too short. Well, he wasn't too short by me, but so I'm not sure. Okay, well, so I stand corrected on that one. But his favorite ice cream. So I have information from different sources. Dominique and Jill suggested that it was blueberry ice cream, but Sandra Deegan suggested it was peach ice cream. But either way, I think he really did enjoy his ice cream. And then finally in the lab, though, this was a unanimous decision. Everybody voted for this. And Ross, do you know what he was called in the lab?
Hanqi.
Hanqi. Is that right? Did I spell it correctly? Okay, fine. So those are very important trivial pursuit answers. I do want to take the opportunity to thank, of course, Mira, Stephanie, Ed, and Christine for all the hard work they did. Really, they did everything. This is much more challenging than you would think in person, quite frankly, coordinating all this behind the scenes. It's truly behind the scenes electronically. Actually, this is a heck of a lot more stressful, quite frankly. For me, I much prefer to do this in person, quite frankly. I don't have to mute and unmute and all those other things. Now, I would like to give the final words to Ross. And then first I'm going to play this. This was in 2013. Eddie Fisher had given a talk on his Nobel Prize winning discovery of reversible phosphorylation. Eddie was a longtime friend, as most of you know, of Earl's. And he's just turned 100 years old just in the last few months. He had a big birthday party. And he used to come with Earl every year to this meeting.
Earl is a very special person who taught us not only to do really good science, but to enjoy it, and that nice people can actually come first. And it's a real honor to see Earl and Anita here again for this symposium. Eddie mentioned Earl's charm, and I really wouldn't be here today without Earl, not because of my scientific work with Earl, but really the US immigration, because I'd come to the United States to train on an exchange visitor's visa, and one day I got a letter in the mail from those friendly immigration folks saying that I would be deported in about 30 days. So I showed this to Earl. Earl took the manager of the Department of Biochemistry to one of the federal buildings in Seattle. As I understand it, he spent 3 hours working his way up to the top, and eventually he came back, and a sign of Earl's charm is that not only was I allowed to stay, I actually got a written apology from the immigration people.
So I think Ross is here and he wanted just to say a couple of words before we really wrap up. Ross?
Can you hear me?
I can hear you and we can see you.
Well, thank you, Ed. I really wanted to say a few words just to thank Ed and his team for actually continuing this symposium. We started it back in 2007 as a way of honoring Earl. And back then we had Ulla Hedner and Johan Stenflo come up and give a couple of talks, which were followed by snacks and beer and wine. So a good time was had by all. We then continued it. This is the 14th one, and Earl came up to every one of them until this year. So we kind of used Earl's name shamelessly to attract the best hematologists in the world to come to Vancouver and share in this symposium. So I also wanted to thank Ed and his team for continuing this wonderful event, even in these strange times. 2007 was also the time that the date that Ed Prysdale and I met Ed Conway in Geneva at the ISTH meeting and tried to convince him to come to Vancouver and become the director of our Center for Blood Research. So I realized with this symposium, this is probably the last one that Ed Conway will be the direct host of. And so I wanted to say a very big thank you to Ed for what he's done for both this symposium as well as the Center for Blood Research. We brought him here because we needed to strengthen our connections to the clinical community in Vancouver. And we also wanted some fundraising, and Ed's personality was perfectly matched to do both of those things incredibly well. So not only do we have the Earl Davie Symposium, but Dana Devine and Ed organized a Norman Bethune Symposium in the spring. We also have big education programs funded by sponsors who are recruited by Ed Conway. So Ed, it's been 11 years, I think, when you've been our leader and also our colleague. So I just wanted to thank you for all the things you've done for the CBR. Through your leadership, your energy, enthusiasm, and dedication, we're in such a better shape than we were when you first came. So from present and past CBR members, thank you so much.
Thank you, Ross. Very kind. Appreciate it. It's all that beer that you gave me in Geneva. That worked. Okay, well, thank you very much. We will see you hopefully really in person in 1 year at the next Earl Davie Symposium, if not sooner. And I encourage you, and I hope you all stay well, stay healthy, and continue to make great, fantastic progress. So thanks again to everybody.
Ed, I'd like to thank you personally as well. This is Jim.
Thank you. Oh, and Jim, yes, this is Jim's, Earl's son. Jim, it's been a pleasure. Great honor, as a matter of fact.
Yeah. And thank you, Ross, for starting all of this.
Thanks, Jim.
All right. Be well, everybody.